demographic rates of organisms. Question number 2, how does heterogeneity within habitats influence demographic rates of organisms? And question 3, how does the alteration of seascapes through anthropogenic activities affect assemblage patterns and processes? So now we're going to look into the first question. And this study specifically addresses how 2 different types of adjacent habitats can influence the demographic rates of organisms on a focal habitat. And so why does habitat adjacency matter? Well, the proximity of one type of habitat to another can potentially strongly influence population and community dynamics. For instance, in estuarine seascapes, juvenile clams that were found on oyster reefs adjacent to salt marsh grass had lower survival rates, while juvenile clams that were found on oyster reefs adjacent to no immediate habitat had an increased survival rate. So this is especially interesting because with climate change and associated increases in temperature, there is a global migration of species on the tropical and subtropical latitudes polewards. And one of these species are mangroves. And here I'm showing you an image of the state of Florida. And on dark green, we can see the mangrove habitat. And notice that it's particularly dominant on the shoreline vegetation in South Florida. While at higher elevations, we see that salt marsh grass dominates the shoreline vegetation. And indeed, there has been an increase in mangrove vegetation along the northeast coast of Florida, especially by the black mangrove Avicennia germinans. So in northeast Florida, there is the Guanatolomato Matanzas National Estuarine Research Reserve, or the GTM Reserve, which is in northeast Florida. And this location in itself is in a transition zone between subtropical and temperate climates. Within the reserve, the estuarine seascapes are composed of beautiful salt marsh grass, as well as oyster reef patches. This location is also undergoing a transition in vegetation. For example, in some areas of the reserve, especially in the south, black mangroves are starting to encroach into the shoreline vegetation. Thus, oyster reefs in this particular location can be found adjacent to different vegetations. And so this is a close-up of the reserve. And we can see that salt marsh can be found in the northern locations dominating the vegetation, while in southern locations, the black mangroves are starting to encroach into the vegetation. So this makes it a very interesting location in order to assess what does this mean in terms of habitat adjacent to the oyster reefs. Which brings us to my study question, which is, are eastern oyster demographic rates influenced by adjacent habitats, such as smooth cordgrass, salt marsh, and the black mangroves? So to answer this question, I had 2 sites north, N1 and N2, where I conducted experiments on oyster reefs that were adjacent to salt marsh grass, and also conduct experiments on oyster reefs that had no immediate vegetation adjacent. And I also had 2 sites on the south where oyster reefs were adjacent to black mangroves, and also oyster reefs that had no immediate vegetation adjacent. And as I had sampled oyster reefs with no immediate vegetation adjacent, these serve also as my regional controls in case there were any differences depending on the seascape location, north versus south. So to address these questions, I obtained juvenile oysters from a local hatchery. As you can see, I attached 6 juveniles per tile with marine epoxy. And then these tiles were assigned either of 3 different levels of predator exposure. A cage, which was completely sealed off of predators, which allowed me to keep track of shell height from the moment of deployment until the moment of retrieval, which allowed me to calculate growth rates. Also, I had the control, which was accessible for predators partially. And I also had a fully exposed tile to allow predator access. And the survival was calculated from these juveniles as the proportion of live oysters at the time of retrieval, which for these tiles was a month later, and for the cages, 3 months. So these experimental units were then deployed at the boundary between the reefs and the adjacent vegetation. Again, just to remind, in the north we had the reefs adjacent to salt marsh, and also reefs with no immediate vegetation adjacent. And on the south, I had reefs adjacent to black mangroves and reefs that were not adjacent to vegetation. And these isolated reefs allowed me to measure predation effects in the absence of adjacent habitats. I also acquired other data sets. I acquired a xanthic crab abundance, and these are predatory crabs of juvenile oysters, such as stone crabs as well as mud crabs. And I also accessed water quality data by the reserve monitoring stations. These data were analyzed with non-parametric permutation-based analysis of variance or through NPT tests. So now let's look at the results. We have on the y-axis survival rate of juvenile oysters, and on the x-axis the location in the seascape, whether it was north or south of the GTM reserve. So we see that in the absence of salt marsh, which are on the black triangle, in the absence of salt marsh or black mangrove as an adjacent habitat, survival rates were higher south of the GTM reserve compared to the north. And these regional differences are consistent with the observed densities of one of the main predators of juvenile oysters, the xanthic crabs, which were higher north than south of the reserve, which could have contributed to lower survival rates of juveniles or oysters that were observed in the north. Now we see survival of juvenile oysters adjacent to salt marsh on the dark circle or in the black circle, and survival of juvenile oysters adjacent to black mangroves on the open circle. And we have some interesting results in that they're opposite effects of adjacent habitats. Survival of juvenile oysters was higher than background rates when oyster reefs were adjacent to salt marsh grass, while survival rates were lower when adjacent to black mangroves. And the reasons behind these patterns are unclear, but reasons could include that there may be a predator associated with black mangroves, which may have an easier access to these oyster reefs adjacent. If we take a look at growth rates, we see growth rates on the y, and again the location on the x-axis. And similarly, like before, we see that in the absence of any vegetated habitat, growth rates were higher on the south of the reserve compared to the north. And this could have been related by higher chlorophyll A concentrations that were observed in the south location of the reserve compared to the north, because it will mean higher food resources for the oysters. Now, if we plot again our juvenile growth rates adjacent to salt marsh on the black and adjacent to the mangroves on the white, we see this opposite effect again, where growth rates are higher adjacent to salt marsh and lower when adjacent to black mangroves. So in summary, for this chapter, we learned that vegetated habitats are important for oyster demographic rates. We saw that oysters adjacent to salt marsh have increased survival and growth, while oysters adjacent to black mangroves had lower survival and growth. So why should we care about oysters? Well, oysters are important economically because they have a fishery value, but also they are important ecologically because they perform a variety of ecosystem services, including filtration of water, which improves our water quality. They stabilize sediments, preventing erosion of other coastal habitats. And they also provide habitats for many invertebrates and vertebrates, which are often of commercial importance as well. But in spite of oysters' ecological and economic importance, oyster reefs are being threatened on a global basis for a variety of reasons. And unfortunately, Florida is not doing any better. We can see that from a 20 to 100% of oyster reefs have been lost in Florida. So in addition to the ecological implications of what it means to be adjacent to one habitat versus another, there may be some broader implications of how oyster populations are going to respond to the continued northward migration of black mangroves. All right, so we just addressed the question of what it means ecologically for species living in one habitat versus another. And now we're going to talk about heterogeneity within habitats. So eastern oysters can form subtidal reefs, but they can also form intertidal reefs. And these intertidal reefs during low tide are exposed to the air, while during high tide, they are completely submerged. And this intertidal gradient of aerial exposure can influence biotic and abiotic factors at different tidal elevations that influences the oysters' demographic rates, or may influence demographic rates. For instance, at higher elevations, these oysters may be exposed more strongly to the physical aspects of the environment, such as higher solar radiation, which increases desiccation stress for oysters. While lower tidal elevations, these oysters may be exposed longer to marine predators, as well as the settlement of other oysters or sessile invertebrates that may compete for space, such as barnacles. But the eastern oysters can also grow in another type of intertidal habitat, and that is on mangrove prop roots. Notice now that we're talking about mangroves and not black mangroves, which I was talking about in the first study. And since this habitat is also intertidal, we could expect that similar biotic and abiotic factors like we just discussed in the reef may also influence oysters on mangrove prop roots. However, mangroves are different from oyster reefs in that they have these structurally complex roots as well as a canopy, which may act as an umbrella filtering some of that solar radiation. Think about like when you're in a very hot summer day and you're having a picnic. If you sit on the sun, it's quite hot, but if you happen to be sitting under a tree, it's less hot. So similar concept. And that canopy of the mangroves may lower desiccation stress for oysters. Which brings us to my study question, which is how do demographic rates vary across intertidal zones for oysters growing on reefs and on red mangroves? So this study was conducted in our beautiful Tampa Bay, here in our backyard. And I chose sites or locations in Tampa Bay, like Upper Tampa Bay Park, Clam Bayou, and Fort DeSoto, because they had oysters growing on mangrove prop roots, as well as on intertidal oyster reefs. To address the questions, I sampled oysters from June through November of 2018. Look at oyster density as well as the density of predators. And I also quantified juvenile survival similarly like I did in the first chapter, with the only difference that I didn't obtain juveniles from a hatchery. I just settled the tiles in the field and kept track of those oysters that settled. And I did this at 3 intertidal locations, such as we see in this image. So in order to sample prop roots, similar areas I sample in reefs, I had to be a bit creative. And first, I went out and did some exploratory surveys to figure out how high on the prop roots these oysters grow. This gave me an approximate length of about 30 centimeters. And then I chose 6 roots with radius of 2 centimeters, right, which will give me a surface area of a cylinder of about 0.24 meters squares. And I did that, and I quantified in 10.5 increments to have that low, medium, and high zone on the prop roots as well. So on the oyster reefs, I use quadrats of 0.5 by 0.5 meters. And I sampled the top of the reef, which was considered the highest point of elevation in the reef. I also sampled the bottom of the reef, which was considered the edge, or where oysters were starting to become sparse on the reef. And middle, which was halfway in between these 2 locations. And the data were analyzed with NP-ANOVAs and NPT tests. So let's look at the results. We have on the y-axis density of oysters. And on the x-axis, we have adult juveniles and total density. So first, on the dark circle, we have densities of oysters on reefs. And we see that adult and juveniles were similar. Well, when we plot the density of mangroves, we see that there were higher juveniles than adults. But most importantly, we see that the density of oysters was higher on mangroves compared to on reefs, and that this pattern was consistent both on juveniles as well as on adults. Possible reasons for these patterns may include the fact that when I sampled for oysters and predators, I found crown conchs, which are a marine predator of oysters here in Florida. And I did not find crown conchs on the prop roots. So this suggests there might be a physical impediment of these organisms accessing oysters on the prop roots. Perhaps the root structure could offer a predation refuge or affect foraging efficiency, but further studies are needed in order to know whether this is the case. In addition, when I deployed the tiles, I also took notes on barnacle presence. And I found that the tiles deployed on oyster reefs had higher percentage of cover by barnacles than on prop roots, which may compete for space with oysters. Not clear why these patterns, so for the research we need to find out. So now we're looking at survival rates here on the Y and intertidal locations, bottom, middle, and top on the X. So again, we see on the black circle those that were on oyster reefs, and while the survival rates did not differ at the 0.05 level, we see this inverted V shape here, or inverted V pattern, which is consistent with our understanding of intertidal ecology of rocky intertidal shores, where lower intertidal elevations are usually controlled by biotic factors like predators, while the upper intertidal might be controlled by abiotic factors such as solar radiation and desiccation stress. And indeed, I found higher abundance of predators at lower intertidal levels. Now, when we plot the survival rates on mangrove prop roots, we see that the survival rates did not follow the same pattern. There were not strong differences between bottom and middle, but there were strong differences between the top of mangrove prop roots and the top of reefs. So it's interesting in particular to focus on this difference in the upper intertidal zone, because if we recall our initial image of this chapter, this goes in agreement that mangrove canopy may be lowering desiccation stress for oysters. But I wanted to look more into this mechanism, and so I conducted an evaporative water loss experiment, which consisted, as you see, on using these small jewelry mesh bags that I filled in with vermiculite, which is a mineral mix, and it's used for potting plants because it retains water. So I fill all these vermiculite bags, with a constant weight, submerge them, weight them, and deploy them on top of the prop roots as well as on the reefs for 2 hours during low tide. And then I collected them again and reweighed them, and the difference in weight told me evaporation of water. And indeed, there was a higher evaporation of water occurring at the top of oyster reefs compared to under the mangrove canopy. And something worth mentioning is that I conducted this particular experiment in February, where temperatures in Florida are not as high. And if we were to, it will be interesting to repeat this experiment during August or in September when it's quite hot, and I'm sure the differences will be drastic. So let's recap. In this study, we found that oyster density was higher on red mangrove prop roots than on oyster reefs. We also saw that the survival rates of juvenile oysters were higher at the upper intertidal zone on red mangrove prop roots. And we also saw that evaporation of water was higher on oyster reefs compared to red mangrove prop roots at upper intertidal zone. So what does it all mean? Well, there's a lot of effort and money put into restoration projects for oysters all around. And the results from this study suggest that red mangroves could serve as a refuge for desiccation stress for oysters. So as temperatures keep increasing due to global climate change, perhaps restoration efforts nearby red mangroves could benefit oysters. And it's also interesting that while we saw a positive effect with red mangroves, we saw a different effect or a negative effect with black mangroves. So although red mangroves have not become the dominant vegetation migrating north, they are also continuing migration with climate change. So that's also something we need to keep in mind. So in the first two chapters, we talk about how one adjacent habitat may influence ecological and demographic rates in another. And we also look at how variation within habitats can affect demographic rates. Now we're going to talk about, in this last chapter, how anthropogenic effects can alter seascapes and influence associated assemblages and processes. So now I want you to take a little break from oysters and think back about that tropical seascape I referred to in the very beginning, in that it was heterogeneous, composed of seagrass beds, as well as coral reefs. And there has been an enormous amount of work in trying to understand connectivity between these habitats in tropical seascapes. And another habitat has been commonly found in these seascapes is macroalgal beds. And these are macroalgal habitats that occur naturally by canopy-forming benthic macroalgae. Compared to temporary macroalgal beds, we are really just beginning to understand their ecological roles. And there's another type of macroalgae habitat that can exist as a result of anthropogenic activities. So in many tropical locations, anthropogenic activities have altered seascapes, and macroalgal farming is one of these examples. The locals farm this macroalgae to export it as a gelling constituent for toothpaste and cosmetic products. But this introduction of farms can also alter the seascape in a couple different ways. As we see in this image, it introduces rows and rows of monocultures. And often this algae that is farmed, the strain is brought from Southeast Asia. So not only it's altering the seascape, this location can also have other benthic habitats such as seagrass beds, bare sand or coral patches, and these farms are introduced into these locations. So they can replace these natural habitats as well. And this is similar or analogous to land agriculture in that it can alter landscapes. We see that from agricultural studies, natural heterogeneity is being lost from these rows and rows of monocultures, which can then affect resource availability, refuge value, resulting in decreased species richness or reduced species richness. So it is unclear how macroalgal habitats can compare to macroalgal farms and the surrounding seascape, especially with respect to how the fish assemblage is reacting to these habitats and how it might affect the process of herbivory that is carried out by herbivores in the seascape. So this brought us to this question, which is how do fish assemblages and herbivory vary between natural macroalgal farms? No, natural macroalgal beds and macroalgal farms, and within the seascapes. So this study took place in Mafia Island, and Mafia Island is located off the coast of Tanzania in East Africa, and it's a beautiful place. And we focused on this location called Chole Bay. And for this study, I selected 3 sites that had 3 macroalgal farms and 3 macroalgal beds. So the survey designed to address these questions consisted on having each focal habitat, whether it was a farm or a natural macroalgal bed, and conducting experiments and surveys in the interior of these habitats at the edge, at 10 meters, 50 meters, and 100 meters. Notice that this is not to scale, it's just to provide a visual representation. And the methods included surveying the fish community using strip transects of 25 by 2 meters. First, I counted and identified all the mobile species of fish. Then we focused on the demersal or more benthic species. And also we counted all the urchins since they are important herbivores in the systems. So every 5 meters of the transect after the visual survey was conducted, I used quadrats to quantify and categorize all the substrate and to get a better idea of the macrophytes that were present, as well as the presence of any hard substrate. And this was done using the design that I just explained in the previous slide. And to quantify herbivory, I conducted a field experiment where I collected algae from natural macroalgal beds and algae from the farms, so farm algae and natural algae. And we remove and clean them off of epiphytes, spin them really fast in a salad spinner to remove any excess water, took their weights, assembled them in a unit such as the ones that we see over here, and deployed them for 24 hours. Afterwards, we collected these macroalgae. We reweighted them again. And the difference in weights was herbivory. And again, we used the same design, and the data were analyzed with NP ANOVAs and t-tests. So what did I find? So we have on the y-axis fish density, and on the x-axis these different locations in the survey design. So first, on the black circles, we have the fish density on the natural seascape, which is composed of the interior of the natural macroalgal beds as well as the different distances. And we see that fish density was similar between the interior and edge of the natural macroalgal beds. While fish density decreased or was higher inside the macroalgal beds compared to the outside. And possible reasons for these patterns include that within the interior or within natural macroalgal beds, there was a higher composition of a higher cover of hard substrate as well as different macroalgae. While in the vicinity, there were habitat patches of a more homogeneous seagrass. Now, if we plot the farm seascape, we see that fish density was lower inside the farms compared to outside. Possible reasons include that in the vicinity of the farms, there were other habitats, benthic habitats present, such as a very long-bladed seagrass, which could have offered higher structural complexity. Now we look at fish density inside the two macroalgal habitats, the farm and the natural. We see that fish density was similar. And this could have been related to similar height and cover of macrophytes in the interior of these two habitats. So we just look at fish density, and I wanted to know how many different species were there. So here in the y-axis, we have species richness, and on the x, we have the different distances. So let's start, just like we did before, first with the natural seascape. We see very similar patterns to those of abundance in that species richness was higher within the macroalgal habitats than outside. And again, this could have been related to the fact that macroalgal habitats or beds were more heterogeneous compared to more homogeneous seagrass patches. If we look at the farm seascape, we have a different result in that fish species richness was consistent throughout. And this could have been related to the fact that there were macrophytes inside the farms as well as outside the farms. So now if we look at species richness in the interior of the macroalgal farms and the macroalgal beds, we see that species richness was higher on the macroalgal beds. And this pattern could have been influenced by the higher cover of hard substrate as well as the other macroalgae and sponges found within these habitats. While in the macroalgal farms, we had rows of monocultures. And if we recall in the beginning of this study, of this chapter, I mentioned how this was similar to landscape agriculture in that it can reduce heterogeneity of habitats affecting species richness. And we're seeing a similar pattern here. So now let's take a look at the results for herbivory. We have now on the Y percent of macroalgae loss, and on the x-axis the farm seascape and the natural seascape. And we see that in the different colors, we have the native macroalgae as well as the farm macroalgae. And higher herbivory was found in the macroalgal farm seascapes compared to those on natural macroalgal beds. And this is quite interesting, and possible reasons include the fact that the fish assemblage had higher abundance of browsers, which are fish species that target macroalgae in their diet. And we had species such as some parrotfishes, as well as the cyganids, which are rabbit fishes, and these are quite common browsers in the Indo-Pacific region. Also, one of the students in a research team, she was looking at stomach contents of these cyganids, the rabbit fishes, and she found that a high percentage of this diet was composed of the farm macroalgae, which suggests that the farms may be attracting these browsers and which may be feeding off this readily available macroalgae. In comparison, on the natural macroalgal beds, there was a higher abundance of invertivores and omnivores, which could have influenced the lower herbivory rates found in these seascapes. So just to summarize, we saw in this study that macroalgal beds and macroalgal farms differ in heterogeneity, fish assemblage, and herbivory. We also saw that patterns of fish assemblage and habitat composition differed across the seascape. And we saw that macroalgal farms attract browsers, altering patterns of herbivory. And some of the implications involve that the alteration of seascape, like we mentioned, can affect habitat heterogeneity, food resource availability and quality, which can result in modifications of food web structure, as well as the associated community and the functions that this community performs. So that was a lot of information. So let's discuss some overall conclusions. So let's recall our landscape image from the beginning, in that landscapes and seascapes are heterogeneous, and that there are many different ways of assessing landscape heterogeneity, from comparing different habitats, looking at within habitat variation, or looking at the effect of one habitat adjacent to another, and even the alteration of seascapes through anthropogenic activities. In this dissertation, I cover all of these approaches. And we conclude from my dissertation, that habitat adjacency is an important factor. And not only habitat adjacency, but the composition of these habitats are important and can influence demographic rates of organisms. We also learned that heterogeneity can occur within habitats, such as variation in biotic and abiotic factors at fine scales, which can also affect demographic of organisms. And finally, we learned that anthropogenic activities can alter the natural structure and composition of the seascape, which can have secondary effects on the associated fauna and ecological processes. So the overarching goal of this dissertation was to use a seascape ecology approach to understand patterns and processes. This work focused on habitat heterogeneity at multiple spatial scales, and I hope to continue using this approach to further understanding of ecological systems. And with that, I want to give thanks to the support of many funding agencies. I want to thank my committee for their support in this dissertation, my collaborators from Sweden, especially Charlotte Bergström, who is probably watching from Stockholm right now. And with her, I wrote a proposal for the Africa work, and it opened opportunities for so many other projects. So that was definitely amazing, and I'm very thankful. I also had lots and lots of help. Like, I could not have done this with so many field assistants. And here are some of them. You saw some of these pictures in the beginning. We have Matt and Owen and Tim, who they were so kind and helpful in my Northeast Florida study. I also had a lot of help from here locally. You cannot tell, but here in the middle, that's my mom. And she's sitting right here. I dragged my mom into a lot of times in the field, and she was a rock star. So I'm super thankful for everybody that helped me. I even had my little brother help me count oysters along with so many other friends. And I was part of this great team of young and talented female scientists that I was able to live with them in Africa, and they are now in Stockholm, in cold Stockholm. But they were an amazing team, and I learned so much from them, so I'm super thankful. And of course, last but not least, I had so much support, emotional support, especially from my parents who are here, and my sister and brothers. I had from one of my best friends, Tasha, and my family from Toastmasters, as well as the best support group that you can ever wish, which is my family here in St. Pete, and a lot of them are pretty much almost here, all of them. And last but not least, my husband, who was the most supportive person every single day, even supported me to leave for 6 months out of the country. So that's pretty good. So thank you all, and thank you for being here today, for hearing my talk.
All right, we're open for questions. Maybe you'd like to field your questions from the audience and from the committee. And the committee can chime in as they get.
Okay. Yes, Sarah?
Thank you for the presentation. In reference to your second chapter, I was wondering if you thought about any of the effects that our lovely sewage discharges may have had on your results. I know it happens a lot. I don't know if you looked to see if there were any significant ones during your study period.
Yeah, that's definitely a good question. I especially thought about the site by climb value, especially when it's lots and lots of rain and everything. I did not look specifically into that, but could have affected growth rates, perhaps, which could lead into survival rates, but I don't have those data to answer that question. But I could expect that will definitely, if it increases chlorophyll in the water and everything, will definitely affect oysters. So, yeah. Yes, Mackenzie?
Do the oysters have any adverse effects on the mangrove growth? I know they're growing on the crop roots, if they're having difficulty taking water because of their growth, or anything
Well, they can help stabilize the sediment, so that also prevents further erosion on the shoreline. So I think it has a positive effect on the mangroves. I don't know in terms of negative effects. I'm not quite sure. Anybody else? Don't be shy. Yeah.
I guess not as much of a question as maybe a comment with the question.
Okay.
Is that you've been saying that black mangroves are migrating north.
Right.
But also the black mangroves in comparison to the red mangroves would be those that are more tolerant to extreme environments, right? They would be the most tolerant to salinity, higher salinity gradients.